Well, good afternoon, everyone. My name is André Drent. I work for QAFI in the Centre for Horticultural Science. And so we have a seminar today, which will go from 12 to 1 by Alistair McTaggart. Can I please have the next slide? I'd like to acknowledge the traditional owners and their custodian on the lands we meet today, and I pay respect to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognize their valuable contribution to Australia and global society. Now, the First Nations people had a very deep understanding of animal and plant life, but also of fungi. And with regards to this seminar, which is about fungi, we know from early records from the early explorers such as Gray from 1841, for example, that the First Nations people use fungi quite widely as a food source, but also for medicinal purposes. They also enjoyed a desert truffle in Central Australia, so they have quite a good understanding of these issues. Can you go to the next slide? So this seminar will run from 12 to 1, but we will go to about 12:45, 12:50, and then we have questions. If you have any questions, please put them in the Q&A tab and not in the chat function. I will then consolidate some of these questions at the end, and you can type them in during the seminar. So today's seminar is by Dr. Alistair McTaggart. Alistair is a research fellow in the Centre for Horticultural Science. He has done some postdocs in the US, in Africa, and in Australia, has worked on rust fungi, has worked on smut fungi, and more recently he started looking at the biodiversity of psychoactive mushrooms in Australia, which is quite a new research area for us, which is very exciting. Today, Alistair is actually going to talk about his favorite subject, which is convincing people that asexual fungi still have sex, and I hope he can convince you during the life of this seminar. Alistair, the floor is yours.
Thanks, Andre. You know what I like. I love it. Alistair here, everyone, and I'd just like to echo Andre's acknowledgement. As a mycologist, I recognize that Aboriginal people were the earliest innovators of fungi. They had knowledge that fungi were sources of biomaterials, foods, and medicines. But today we're looking at sexual reproduction. And pretty much this is work that I was doing in COVID through peaks and troughs of boredom and depression or whatever. And it wouldn't have happened without lockdown as the catalyst. And it also wouldn't have happened without all of my co-authors who just tolerate me and listen to my emails or read them and put up with my eccentricities. So this week, yesterday, I saw an email sent out from UQ saying, "What's on at UQ?" And I saw a well-lit photograph of some magic mushrooms. And I thought, oh, wow, I got to check this out. What's going on? And it turned out it was advertising this seminar. Oh, flagrant false advertising. This, for anyone who's here to hear about magic mushrooms, this is going to be a bit of a bummer. This is about fungal sexual reproduction. So, you know, everything that I'm going to talk about is what I'm doing with magic mushrooms. If you really want to hear me speak, I'll be talking next Wednesday at Entheogenesis Australis seminar on Wednesday night with a Q&A session. And I'm making sure that everything that I do is really transparent. You can follow my blog or look at my blog periodically, and there'll be updates on the magic mushroom research as that goes on. It is one slide about magic mushrooms. It is, with all this rain in Southeast Queensland, it's such a good time to be collecting fungi, especially magic mushrooms. And on the weekend, I had the opportunity. That's me, and this is Chris Appleyard, the founder of Funky Fungus. They're the first company with permission to start producing psilocybin in Queensland. And we were on his property, and that's a Psilocybe cubensis in front of us. And hopefully that might be the start of something special for him with getting that into culture, and he can start producing magic mushrooms for clinical use. So, yep, we're not talking about magic mushrooms here. We're talking about sexual reproduction. And now that it's just, the people that care about sex see that the numbers have dropped down to about 6 of us enjoy this. We're going to have a good time. So fungi are such good pathogens because they can reproduce clonally, asexually, and sexually. And they're so good at clonal reproduction that it tricks mycologists and plant pathologists all the time. We'll, sample from epidemics because that clonal stage is just reproducing that genotype, and it's really cool because it keeps the perfect combination of alleles and it proliferates. And so we'll sample from that and we'll go, everything's the same. It mustn't reproduce sexually, or it's lost the ability to reproduce sexually. And this has happened for lots and lots of different agricultural and human pathogens. And so in COVID, with surplus time in the evenings, I tried to answer a few of these questions about clonal fungi, and see if they have sexual reproduction. And so you're just going to hear 3 stories, one about Fusarium oxysporum, which is a predominant clonal organism, one about Quambalaria, which no one had any idea of what was going on in that life cycle, and then one about Myrtle rust, which is an invasive organism in Australia, and it has the best of both worlds, clonal and sexual reproduction. So Fusarium oxysporum is kind of a big deal. Just within coffee, some of these names might look familiar. Fusarium oxysporum, often you'll see a little F.sp, which means forma specialis, followed by a name. And that name is just an indicator. It has no taxonomic rank or value. It's just an indicator usually of the host that it was taken off. So Andre works on cubensi, which is the Fusarium oxysporum on banana, or you'll see vas infectum is the people working on Fusarium oxysporum on cotton. And if you work in agriculture on a plant, you probably have Fusarium oxysporum on your crop. Or if you don't, Fusarium oxysporum is 100% in the soil beside your plant. It is really, really common. Okay, a little bit about the background of life cycles of fungi, because I love life cycles because they're linked to sexual reproduction as well. So with most ascomycetes, including Fusarium, ascomycetes hang out a lot. The majority of their life cycle is as a haploid. And by that, I mean there's one nucleus and one copy of each chromosome. And these asexual stages, which are conidia in ascomycetes, they infect something and they produce the same genotype and produce more conidia of the same genotype, and the cycle goes on until they meet a compatible partner when they can anastomose. And there's a brief period where there's 2 nuclei. So there's 2 copies of each chromosome, but in separate nuclei. Those nuclei fuse in karyogamy, and you have meiosis pretty much immediately following, and the production of a recombinant spore, an ascospore, which then causes an infection that starts producing conidia again. Spine tingly dingly. That's pretty applicable across most ascomycetes. In Fusarium oxysporum, there's never any anastomoses. And these kind of different mating types, the ships in the night, and this is the hypothesis, and they never cross, even though it's pretty common knowledge that there's a good distribution of MAP1.1 and MAP1.2 in populations. The hypothesis is that there is no recombination. And the taxonomic community, or the Fusarium oxysporum community, they don't really care about sexual reproduction. At the moment, there's this huge taxonomic tussle going on. Some people think that there's less biodiversity in Fusarium oxysporum. Others think there is more biodiversity in Fusarium oxysporum. So you'll have people who say it's a species complex of 3 species, or it's a species complex of 10 or more species. And this taxonomic turmoil has left about 500 genomes on GenBank. So it's perfect for a little data parasite like me to come along. Thank you to the taxonomic turmoil and people not asking whether there's sexual reproduction. It means that there's a whole bunch of data for me to look at, and no one's ever touched this question of whether Fusarium oxysporum completes a sexual life cycle. So I'm just looking for the stuff that everyone looks for to test if something's having sex. So is there an equal distribution of mating types? do all the trees, when you make a phylogenetic hypothesis of genes, do they all support the same thing? That wouldn't be sexual reproduction. But if they all support something else, different relationships, that is sexual reproduction. Reticulation in networks. I explained this really poorly. I'll come back and explain that again. Sorry. So reticulation in networks and rejection of pairwise homoplasy, and then also rejection of linkage disequilibrium. So if you're the type of person that likes to see what tools I used, I pretty much downloaded 500 genomes, annotated them with Augustus. That number came down to 410 genomes after I filtered them for quality based on their completeness of annotated genes. We were really only working with the core genome. We were using single copy orthologs identified by OrthoFinder, and we called SNPs with KSNP, which is a Kama SNP calling program. And sorry, I should just a little bit of background for our sugarcane comrades. Fusarium oxysporum has a 50 million base pair genome and it's haploid. So super easy to work with compared to something like sugarcane or any diploid. With KSNP, we're just calling Kamas from those haploids, and you set the Kama value, and then we were only calling a SNP site if it was present in every single genome, that locus, if it was present in every genome. So some time estimation, any phylogenetic hypothesis with IQ tree, all our neighbor nets with splits tree, our VCF analyses is with Plink, and then Popper for all our tests for linkage disequilibrium. And then we were looking for repeat induced point mutation, which is active in recombination, identifying TEs with RIPET, and then using RIPCal to identify RIP. Okay. Gosh, it's a bit text heavy. Let's just skip over that and go straight to the meat and veg. So we had two data sets, our phylogenomic data set, which is our OrthoFinder analyses, which is of genes, and then our population genomic data set, which is our SNPs. So our phylogenomic data set, we were using to kind of find structure in this huge population of Fusarium oxysporum a priori. So we were just looking to see if there was anything we could start to delimit. And it turns out there was. So we were able to just kind of get these phylo groups. What I've done is put species names on them. And this is based on some of those people that have a taxonomic hypothesis of more biodiversity. It was really easy to apply these names. And then the phylo groups are just these numbers. We used something called a genealogical concordance factor to support those phylo groups. It's not new, but the community is still catching up to understanding it. So if you're in the mycological community and you haven't heard of genealogical concordance, it's really just the percentage of genes that support a particular node. That, like a bootstrap has an agreed upon value. If you're doing an ultra fast bootstrap, 95% and above is well supported. With a genealogical concordance factor, it could be dependent on the data that you're looking at, because you can imagine if you've got a whole lot of genes, but there's no resolution at like the crown or at certain nodes, so you might have polytomies, then you won't have support. And so that large number of genes the percentage is slowly decreasing. So it's really dependent on data set. We used a cutoff of 20% of our single copy orthologs recovering a node to start calling a phylo group. Each of these nodes, except for some in the crown, had 95 to 100% bootstrap support. So genealogical concordance factors, I love them. And that's what we were using. This is also a time estimate. We calibrated this node here to 5 million years, just based on a clock-like rate of speciation in Fusarium and a couple of other studies on Fusarium. Ultimately, all of these phylo groups, or if we dare, species, have evolved in the last 150 to 250,000 years ago. And this node had a maximum age of 1 million years, but a mean age of about 500,000. So this has all happened in the last 500,000 to million years. We expect some recombination to be going on. This is also using our 40 single copy orthologs. So this is just another way of visualizing data using a splits tree. In splits tree, when you have a lot of reticulation, the splits trees show all potential relationships among data. And when you have a lot of reticulation, you might interpret that as evidence for reproduction or recombination, or you might interpret it as evidence of homoplasy or incomplete lineage sorting. We've interpreted it as recombination because it's supported by other evidence. So each of our phylogroups is colored differently, and then you've got circles and triangles, which is just mating type. So in a well-sampled phylogroup like phylogroup 1, you've got near equal circles and triangles. In 5 and 9, you do as well. But in things like phylogroup 7, you've only got circles. Phylogroup 8 only has circles. Phylogroup 6, where is it? Oh, it has 2 triangles. So you've got, overall, you've got a near equal distribution of 1-to-1, but you do have some phylogroups that only have one mating type. The thing that we were testing with splits tree is pairwise homoplasy as well. Pairwise homoplasy is taking a window across your data set. We had about 50,000 amino acids in our data set of these 40 single copy orthologs. And it's just randomly looking, it's looking at whether there is a random combination across your genomes, okay? A random combination of, say, SNPs across your genome or genes across your genome. I shouldn't say genes, alleles in this case. And you would say that below 0.05 is well supported for reproduction. Above 0.05 is considered probably clonal. So those phylogroups that had one mating type were often had high support for clonal, but usually we rejected a pairwise homoplasy in that data set. So yeah, you've seen that there's a 1-to-1 ratio. So mating, that 1-to-1 ratio was pretty strong for us, that there must be some sort of thing controlling that equal mating type distribution across the populations. In a haplotype network of MAT1, we see MAT1 is pretty diverse, and there's only one haplotype that's shared among the phylogroups. In MAT2, you've got less diversity. MAT1 and MAT2 control a cascade, so a big pathway of recognition or mating recognition, pheromones and receptors. So just because they're the same doesn't mean that everything in that pathway is either. Okay, so our SNP data set, pretty much we called 4.2 million SNPs. That's what we were working with across the whole data set. Each phylogroup, we had fewer SNPs to work with, but still well over 100,000. So we were looking again for reticulation with a splits tree, and then we're looking at the standardized index of association, the RBARD, which, similar to pairwise homoplasy, is looking at a random combination of alleles in your data set. So are things always going together? Do you, at 2 loci, do you always get A and A, or do you get A and T, T and A across the whole data? And as RBARD, or the index of association, approaches 0, you have more support for recombination. So our splits tree analyses of 4 million SNPs return a very similar topology to our phylogenomic hypothesis. Lots of reticulation going on. What's interesting is that we'll have reticulation within phylogroups, and you can see in phylogroup 1, which had a cutoff of 27 for our genealogical concordance factor, there's probably some substructuring. We have no idea of what. But what's important to take out of this figure is that in a splits tree, we would expect, if there was recombination among different phylogroups, is we would expect connections higher up in towards tips among different phylogroups, which we did not see. So what we're supporting is recombination within, but not among or between different phylogroups. Okay, and then just this is a test for pairwise, for the index of association, or RBARD. This is one way to visualize it. So as RBARD approaches 0 in all these triangles, you'll have a purple color. So our really well sampled and mating types that were 1-to-1 in phylogroup 1, 5, and 9 all had this RBARD that approached 0. This is just across 10,000 random SNPs. This much more boring table is a bit more informative if you've got the patience to listen. So we clone corrected across our more than 100,000 SNPs in each phylogroup, over 1 million in phylogroup 1, for instance, and we see a magnitude of difference in the R-bar-D after clone correction, except for phylogroup 6 and phylogroup 8. Phylogroup 9 also didn't have a magnitude difference after clone correction, but it did have quite a low R-bar-D anyway. The last test that we did was to determine RIP, so repeat-induced point mutation, and RIP just acts kind of to keep your genome safe against transposable elements. So it goes through GC-rich transposable elements and changes Gs and Cs to As and Ts. So we took the best assembly based on N50 from each phylogroup, pulled out homologous TEs, and then just looked, this is just using the RIPET pipeline, I should say, and then just looked for activity of RIP. So the more squiggly these lines are, the more RIP activity you have. And there was RIP activity in every single phylogroup. So all of those data or all those analyses support that recombination has occurred in the evolutionary history. We had segregated mating types, non-concordant topologies, we had reticulation, rejection of pairwise homoplasy, we rejected linkage disequilibrium, but in 6 and 8, it was a little bit questionable. And we had RIP activity and transposable elements. And so just because you don't reject linkage disequilibrium doesn't mean that there's not recombination. It supports something else, which is well known in the community that work on clonal organisms as predominant clonal evolution. And to save you reading all of that, long story short, there are just many more clonal generations than there are recombinant generations. And it's a really good way to stop recombinational load and to keep good genotypes going. So that paper should be published really soon. It's been accepted. The reviewers did not like that we had only looked in the core genome. They suggested that we maybe have missed some recombination among phylogroups in the accessory genome. So it's set up for our next study, which I'm looking forward to doing, and it's underway right now. This is a core genome, just single copy orthologs. This is over 3,000 single copy orthologs, and it just recovers the exact same topologies that we've seen before. This is a big network, and it's the map for me to look for horizontal gene transfer. So I'm really just looking for horizontal gene transfer among the accessory genes and chromosomes. So Fusarium has dispensable chromosomes that sometimes carry genes that help with pathogenicity. This little network here is made from over 2 million accessory genes, and it just recovers the exact same phylogroups as what we had from the core genome. And I'd like to invite anyone that wants to work on horizontal gene transfer to come and help with some of these data. So we're looking for patterns. I might explain on this slide. So we're looking for conserved gene order that occurs in different phylogroups, but not in every individual of those phylogroups. So that's how we're identifying horizontal gene transfer. In this case, phylogroup 6 always has this order, phylogroup 5 has this order, then you get down to phylogroup 1 and there's been an inversion and something's been swapped around. So these genes, there's 2 genes in this 12 to 20,000 base pair region. Locus, it's only present in these 13 genomes of that 410 genome database or data set. So pretty cool. Get involved. Email me if you want to help. So story number 2, better just check the time here. Cool, looking good. Quambalaria. So this is work with DAF, and in particular, Jeff Pegg, who is working on Quambalaria in his PhD. He had a data set lying around, and he'd given it to me because he doesn't have time to work on this kind of stuff now. And COVID was the perfect excuse in the evenings to start mucking around with Jeff's data. And I think that's a lesson, even though, no one asked for my opinion, this is it anyway. You get to hear it. Your colleagues are sitting on untold amounts of data. If you ask them and you're an EMCR or HDR, it might give you something to learn a new skill or to work on in the evenings, if that's the kind of giggle that you enjoy. So Quambalaria, in 2000-ish was shown to be a smut fungus. And, you know, I love working on smut fungi. It's related to the fungus that causes dandruff, malassezia. So it looks like snow on like a fungal snow on leaves of eucalyptus and corymbia, leaves and fruits. And for the longest time, these were considered to be conidia, so the asexual stage in an ascomycete. And it was a big question of what the hell was going on in its life cycle. So in a normal life cycle of smut fungi, so different to ascomycetes, Basidiomycota spend most of their life cycles with a dikaryotic 2 nuclei with, so 2 copies of each chromosome, but in 2 separate nuclei of their hyphae or their spores or whatever they're resting in. So they're most of the time as a dikaryon. Then just before meiosis, you obviously get karyogamy and the production of these recombinant basidiospores. And what's super cool in smut fungi is that those basidiospores just start budding like bacteria. They're called a budding yeast. So it's just mitotic division, and they just grow in chains of spores. In smuts, that stage, so that stage that's clonal, so this is the just mitotic growth, this is non-pathogenic, so it can't infect a plant. It needs to meet a compatible partner and then get 2 copies of a genome or 2 copies of each chromosome so that it can become pathogenic. How interesting. So for this reason, smuts aren't the best causes of epidemics. They just don't have a pathogenic clonal stage. Smuts are a big deal though, because, you know, in a monoculture of grasses, smuts mostly infect the grasses and they'll destroy the inflorescence. So in something like sugarcane, where you have all of your cane stunted by smut fungi, or in wheat, there's a smut called Telesia indica, carnal bunt, and small amounts of smut spores. So even though it's not an epidemic outbreak, small amounts of the spores get in with the grain and they've got a trimethylamine smell, essentially a smell of rotting meat. It's the reason that gingerbread was made. If that's new knowledge, how groovy are smuts? So eventually, yeah, when it's pathogenic again, it replaces the inflorescence with that powdery brown mass of spores. So we have no idea what's going on in Quambalaria. It's just known from these structures, which have always been called conidia. And when Jeff was working on this, back when it was super hot and cool to be working on it, it was causing epidemics. So untold of in smut fungi. So it was causing epidemics on plantations of spotted gum in about 2008, 2009. So Jeff had this big data set and I thought we could ask this. Does Quambalaria have sex, or were these epidemics caused by a clone? So Jeff had already done the sampling about 11 years ago, 12 years ago. He had 177 individuals from 3 different locations. Someone had already done all the AFLP work, all the hard stuff, and I just got given the data. And I'd never worked with the AFLPs before, so it was a new experience and heaps of fun. The first thing I did, as you would do with any population study, is look for structure. And I was looking for structure for a population structured by host or location. So we did find structure, but I have no idea by what. It's not by host and it's not by location. But what is kind of cool is that the data show that there is absolutely really high genotypic diversity. So there's 0 clonal reproduction. Every single individual that Jeff had isolated was a different genotype from others. So we had high genotypic diversity, and then there's that R bar D again. So as it approaches 0, more chance of recombination and rejecting linkage disequilibrium. And because we're not dealing with millions of SNP sites, we can get a p-value as well. So as that p-value goes up, it adds support for rejecting linkage disequilibrium. Plus all this reticulation, just showing that there's so many different ways each genotype is related to the other one. So this is just painting a picture of sexual reproduction, but we still have no idea what structures those 3 populations. So to me, I thought it was a super cool story because it's a smut fungus causing an epidemic. And what's more, it's causing an epidemic in a native pathosystem. So Quambalaria and species of eucalyptus and corymbia are native to Australia. Normally, after millennia of coevolution, they're at a nice equilibrium. When you put them into an artificial plantation setting, then this is where you get that epidemic potential, even though we didn't have clonal spread. So I thought that was a super cool case study. And I'm not aware of another one of an obligate organism kind of causing this thing in a native pathosystem. Okay, the last story is rust fungi, and rusts they're a bit different to smuts. Their clonal stage actually causes an infection. So that smut haploid stage can't cause an infection. The rust clonal stage is a dikaryon, and it's repeating. So that genotype can really start to get some momentum behind it and cause epidemics, which is what happened in Australia with myrtle rust when it came over here in 2010. And it is a biological atrocity. We've got species that are localized, extinct locally, and then we've got species that were already endangered, like species of Lamwebbia, that are now on the brink of extinction thanks to myrtle rust. We had one genotype come in and spread by uridinospores in Australia. If we had another incursion of a different genotype and these things could outcross, then they might be able to overcome the current resistance that's present, although there's little resistance present in our native Myrtaceae. But whatever's left, they might be able to overcome. Myrtle rust may be able to overcome that resistance. And so you can imagine, just based on Quambalaria, from what we were talking about, Quambalaria and eucalyptus had millennia of coevolution. Myrtle rust, Australian Myrtaceae has two rusts on this huge diversity of taxa. They had no idea what a rust was, and then myrtle rust came in and ruined their rust-free party. So when we're talking about rust fungi, mycologists and plant pathologists pretty much are always thinking about the rust that is the most widely studied in the world, and that's Puccinia graminis, wheat rust, very popular rust to study. It is a rust of a temperate grass, an annual, so the host dies, so the rust has to change hosts during its life cycle to complete its life cycle. I'll just take you through it super quick. So uridinospores are that mitotic stage. One uridinospore causes an infection of the same genotype to produce the exact same genotype, uridinium, and more spores. So building up that spore load. A trigger, whether it be environmental, from the host itself, from the rust, we don't know what the triggers are really, will cause karyogamy and the production of teleospores, where there's rapidly after meiosis, or depending on whether they're used for overwintering, they might spend their life in crumpled leaves and then produce recombinant basidiospores. In Puccinia graminis, on wheat, those basidiospores don't infect wheat, they infect a different host, barberry. They cause infection, which produces these structures, spermagonia. A spermagonium is just amplifying the haplotype from the basidiospore. So it's producing hundreds to thousands of these little spermatiospores, spermatia, that are just spreading that genotype to fertilize other spermagonia. Then you'll have fertilization and that production of a dikaryon, because this is a basidiomycota, so most of its life cycle is dikaryotic. And the structure produced by wheat rust is an acium, and these aciospores are just to get from barberry back onto wheat and to kick off that genotype on wheat. Too easy. In myrtle rust, we're trying to apply the life cycle of Puccinia graminis, or they were trying to apply it. So in myrtle rust, you've got this really clear uridinia, and then chalk and cheese in the field. These are telea, these dark brown things, and these telea germinate readily to produce basidiospores after recombination. And because the community had never found acea or spermagonia, they were like, well, maybe there's an alternate host that we haven't found, or maybe it doesn't reproduce sexually. And then there was kind of this trend to conclude that it didn't reproduce sexually and it was a strictly clonal organism. So back when I was living in South Africa, my partner Louise and I, we wanted to test that hypothesis a little more. We separated the gametic spores, basidiospores, haploid spores, from the mitotic stage, uridinospores. We inoculated these onto a susceptible host. We knew the genotype at 6 microsat loci that were heterozygous, essentially. We were doing a dihybrid cross, We just expected a random association of alleles in the F1. And that's exactly what we saw, with other knowledge as well, we thought, okay, there's pretty good evidence of sexual reproduction in myrtle rust. We were able to put forward a new hypothesis for the life cycle, which doesn't include spermagonia and assia. The gray area of the life cycle for the community is what happens when basidiospores meet each other on the leaf surface. So I've put forward hypotheses, but they've been rejected whenever I put them forward. So this is a DAPI-stained basidiospore, and each of these has 2 nuclei. So I think that 2 nuclei migrate in this polar. The basidiospores germinate polar ends, and I think a nucleus would migrate down either, and then you could have that one haplotype could fertilize 2 other basidiospores in the right conditions if they met. So the big question is, oh, hold on, I think I might be jumping the gun for big questions. I am. Let me just quickly give you a little bit more information. The community has known for a long time that the myrtle rust, or Austropuccinia psidii, in its native range is structured by host. So people would take spores from eucalyptus when it was introduced to Brazil. They would try and inoculate guava, and they couldn't. And they couldn't put spores from guava, which is in Myrtaceae. They couldn't inoculate eucalyptus. So they suspected it was structured by host. And then in the 2000s, a Brazilian group showed, yes, Austropuccinia psidii is structured by host. And this is where that big question is. So this is the pandemic strain, which is spread through Australia, New Zealand, and all through the Pacific. And then South Africa had an incursion about 6 years ago. And the thing about these two rusts, well, I shouldn't say 2 rusts, they're the same species, but maybe 2 different strains is one way to think of it. They're polyphagous. They're not structured by host. There's no way to delimit them by the host that they occur on. So the big question for me, and I think the community, is if these strains were to ever meet on a universal host, something they can both infect with their basidiospores, the structuring that was previously by host, that's out the window. Can they now outcross? And to answer this, we have to get into the hypothetical zone, much like a teenager, of sexual reproduction. Okay, yeah, so hypothetically, can different strains of myrtle rust outcross on a universal host? And this is just a photograph of the damage that myrtle rust is doing to our mid-story secondary growth rainforest. This is down in the Talabudra Valley. And with Jeff and Louise, we've been doing plots. 90 out of more than 100 trees in each plot are usually dead from Austropuccinia psidii. Okay, a little bit of knowledge about how Basidiomycota reproduce sexually. This figure is adapted from some leaders in the field of sexual reproduction in fungi. Basidiomycota reproduce either as bipolar or tetrapolar mating systems. In bipolar, the genes for compatibility are all at one locus. In tetrapolar, the genes are at 2 loci. And I'm going to explain this more. When we're talking about genes, we're talking about HD genes, which are homeodomain genes, and pheromone receptor genes. So in a bipolar taxon, this is Sporosorium scidominium, sugarcane smut. This is considered all one locus because it's within 50 kb of each other. So our HD genes, our homeodomain genes, and our pheromone receptor genes are always inherited together. So we consider that bipolar. If you lost, so for Eustilago hortii, which is the type of all smuts, barley smut, they've lost pheromone receptor genes altogether, and they're an easy example of bipolar mating. So tetrapolar mating occurs in corn smut, Mycosarcoma matus. So the homeodomain genes are located on chromosome 1 at that homeodomain locus, and the pheromone receptor genes are located on a different locus on chromosome 5, so they're not linked. And the key to understanding mating in Basidiomycota is that the pheromone receptor, or if you're looking for a compatible cross, you have to differ at your different loci, if you're bipolar or tetrapolar. In rust fungi, we know that there's a homeodomain locus, and we know that the pheromone receptor locus has 3 homologs of a STE3 gene, which is just a pheromone receptor gene. We have a better understanding now than when this figure was first published 2 years ago, 3 years ago. Rust genomes are usually highly fragmented assemblies just because they're so repeat rich. With long read sequencing, they're able to get a better picture of whether these loci are actually linked, and they don't seem to be. So it's a candidate for tetrapolar mating, but let's keep that in your minds. So I've just looked at the HD1 and HD2 genes in Austropuccinia, and we had 3 genomes available, one from our colleagues in Sydney who have assembled the genome of Myrtle rust here, one that we'd assembled in South Africa, and then the colleagues from Brazil who this manuscript was in collaboration with, they've assembled one from Brazil on eucalyptus. And so our HD locus with these 2 genes, each of those genes has 2 alleles in each genome. Okay, so to have that cross, those HD genes had to differ in those 2 nuclei to come together. I hope I've explained that well enough. Whereas our STE3 genes, so our 3 homologs of STE3, you can see based on this phylogenetic hypothesis, where branch lengths are informative in this phylogram, these STE3 genes, even though they're present in Austropuccinia psidii, they're completely identical in the 3 genomes. When we have a look at the STE3 loci themselves, they're highly identical. This is the shortest one, but also the most variable, STE3.2.1. Not that it matters, it's just one of the STE3 genes. And all of the SNPs occur either outside of the gene itself in non-coding regions, in introns, and then there'll be synonymous substitutions if they're occurring in an exon. So yeah, along up to 8,000 nucleotides in a contig, and you're having 99.9% pairwise identity at the STE3 gene. So they're not variable, whereas the HD genes are variable. What does it all mean? It means that myrtle rust is most likely bipolar. So that STE3 homologs, they're not involved with compatibility. They may still be there for mate recognition, but they're not involved in compatibility. They're bipolar. So you only have to differ at the HD locus for a compatible cross. And there are numerous advantages to bipolar mating. Tetrapolar mating reduces your chances of reproduction just because you have an extra locus that you have to differ at, right? Whereas bipolar, you only have one locus that you have to be different at. So tetrapolar mating is there to stop inbreeding. But when you've got wind-dispersed spores, hopefully inbreeding, the chances of inbreeding have been reduced. There are numerous examples in Basidiomycota of the STE3 gene kind of getting taken out of tetrapolar mating. So if they were at 2 loci to begin with, the STE3 gene may no longer be involved in compatibility. So these are 3 mushrooms, for example, but there's more. Okay, so yeah, oh, hold on, let me go back. So my take on this, interpreting this, is that based on the HD locus, technically different strains of myrtle rust should be able to outcross on a universal host, which doesn't bode well if there's ever an incursion, because it means that our strain here can probably outcross with a new introduction. Hopefully, that hypothesis will never be tested. Okay, so from however long I've been speaking, the take-home messages are that when you hear someone say that a fungus is strictly clonal, that's setting you up to test a hypothesis that they're reproduced sexually, which is what we did with Fusarium oxysporum, Quambalaria, pitereka. No one knew really what was going on. And Myrtle rust, even though we've shown sexual reproduction, now I think we've put forward a pretty good case that different strains can outcross. So again, my opinions that weren't asked for, all of these data except for the Myrtle rust data, which are largely mine. So the Quambalaria was given to me by Jeff, and Fusarium oxysporum is totally mined. There's just so much there if you've got the right research questions to ask. My experience with that Fusarium work, I invited one of my mycology heroes onto that paper just so that we could work together. And it was the most fun, just getting another perspective. It was such a great way to collaborate. And I encourage you all to do the same thing. All right. And remember, if you want to get involved with horizontal gene transfer in Fusarium, then shoot me a little email-y. Okay. Andre, back to you.
Thanks, Alistair, for that illuminating seminar. So if you have any questions, please put them in the Q&A section, not in the chat function, and then can ask them to Alistair. Now, Alistair, I'll start off with a more generic question. Is there actually an alternative use for mating genes outside the mating system, and genetic recombination leads to more diversity, blah, 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 blah. But are there any theories for alternative roles for actually getting rid of pathogens or other functions the mating system may have?
Yeah, okay. Andre, I just want to diverge on that question. So because of magic mushrooms, so every magic mushroom I get, I'll sequence the genome of and start to cross, because I'll be sequencing haploids. So I'll be using mating genes specifically to direct my crosses. So instead, back in the day, you would just try and cross everything with everything else. I will be just crossing the ones I know that will already compatible. In an agricultural system, you would know better than me. I can't think of an example off the top of my head. We stop sexual reproduction in rust fungi to try and reduce their ability to change. So just sexual reproduction is making evolutionary innovation, and that's why you don't want it to happen if you're managing plant diseases. But yeah, I can't think of a real example of using a mate type. Do you have one? Maybe something from NAR. Has NAR with
I got a few other questions for you. One of them is from the Tracy Steinracken from the Australian Mycological Society, which is a very interesting question. It goes like, she's wondering how your sexual reproduction bombshells have been accepted in the mycological world.
Yeah, a good question, Tracy. I think that there's a large community of people who support that there's sexual reproduction, and in every fungus, there are pockets of taxa where you've got, and I'm going to quote Andre here, some hypotheses don't die until the people who hold them die. Oh, wait, was that tasteful? I shouldn't have said die. Megan, edit that out. Yeah, so you've got recalcitrant people who will say that there's no sexual reproduction. I would be interested in testing those hypotheses for everything. If there were enough data, I wouldn't do the work myself, but I'd always be happy to ask the question and use those data. For the Fusarium work, that was a bit of a mission to get that through. I think because it had a loaded taxonomic outcome as well, because really the names that you apply to those phylo groups are going to be species, and you would probably apply them based on recombination within them and not among them. That's my take on what I would do with that taxonomic information. But I don't work on the taxonomy of Fusarium, and nor do I ever want to.
That's interesting, because you had another question about that, whether you support the splitting of Fusarium oxysporum. You may want to avoid that question.
Yeah, I think that question is, how long is a piece of string? Applying names. What's interesting is that the way it was split, it's still not informative. So the people who work on banana, TR4 or tropical race 4 and TR1, there's still no way the names still aren't informative for those particular taxa. So those phylo groups, or Fusarium odoratissimum, is not specific on banana. So this is where that accessory genome, and I think the Fusarium community is really interested in that accessory genome for all those kind of things. But you can still apply those names based on recombination, which I think should happen.
Okay, so we have, I think we've exhausted the question. So thanks very much for your great presentation, Alistair. And you have a few complimentary comments I will share with you after the presentation. Can you move to the next slide?
Absolutely. That
So,
blog link, sorry, Andre, that blog link, Eduardo, that can be found on my UQ researcher page.
okay, yeah, so the Weebly page is there. So next week we have a hybrid seminar. So you can either turn up in person or do it online. So please don't forget that. And it is by our well-known Professor Robert Gilbert from CNAFS, and he's going to talk about complex brands, glucose polymers, and their roles in human well-being. So you will get some emails about that, or please see the QAFI website seminar for more information. So in closing, I'd like to thank you for attending this online seminar today. If you enjoyed the seminar, please do the poll at the end. And if you haven't already signed up for our regular invitation list, please do so at quafi.uq.edu/science seminars. Thank you very much, and I'll hope to see you next week. Thanks. Bye.